I'm pleased to introduce our first speaker opening the webinar, Dr. Alexandra Benchoua. She will talk about her journey toward the use of alternative to animal model in neuroscience with human pluripotent stem cells. So as for her background, Alexandra Benchoua trained as a neuroscientist in France with additional formation in biotechnology and in developmental biology in the UK. She is currently a research director at the iStem Institute at Corbeil-Essonnes in France, which is the Institute for Stem Cell Therapy and Exploration of Monogenic Disease, and actually one of the biggest institutes dedicated to stem cell technologies in Europe. She is currently leading 2 teams, so the neuroplasticity and therapeutic team, and one on the research and innovation in technology. And the 2 teams are combining their skills to create new models for neurodevelopmental disorders based on human pluripotent stem cells that are amenable to miniaturization, automation, and high-throughput drug screening. So I will now turn the presentation over to you, Alexandra, and we are really looking forward to it.
Thank you.
Thank you.
So I hope everybody see my screen properly. So today,
Yes.
I have the pleasure to introduce this very important workshop on alternative method to animal testing in neuroscience. And so the introduction will be more on a journey from using rodent fetuses to human pluripotent stem cell to create model in neuroscience. So thank you for the nice introduction. So actually, I'm working south of Paris in a town called Évry at the Institute of Stem Cell to Treat and Explore Monogenic Disease, so ISTEM. And I'm mainly interested for decades now in studying and trying to find treatment for genetic neurological disorder of developmental origin, which is the so-called neurodevelopmental disorders. And I'm also interested in repositioning a marketed drug to try to find a good cure for those neurodevelopmental disorders. So just to give you quickly a figure, there is actually 4,000 compounds on the market that are used for a diversity of disease, and 300 are more dedicated to CNS disorders. So this is the kind of chemical collection you can investigate for genetic disease. So the main question that we had regarding the modeling and the study of neurodevelopmental disorder in vitro and to drug discovery is how to fairly model something that happened in utero, which is the brain development, knowing the fact that neurons are very particular cells with particular morphology and that function as networks where cells communicate with each other using electrical transmission and chemical communication via neurotransmitter and synapses. And knowing also that different type of neurons perform different type of function, and that you can model one type of neurons with another type of neurons or with other kind of cells. So of course, to obtain developing neuron in vitro, you need a resource. And one resource that sounds quite logical would be to try to pick up human neurostem cell to develop neuron in vitro. But of course, for evident reason, there is a big problem of sourcing for brain stem cell. So they exist in adults, but they are located in very discrete locations in the brain that are very deep in the brain. So it's really impossible to collect the cell from an adult, even post-mortem. So we sometimes use fetal brain stem cell obtained from fetuses after abortion, but also for evident reasons, the resource is very rare and not very consistent because, of course, after abortion, the quality of the fetus is not very equal. So for decades, we model the development of neurons in vitro using neurostem cell from embryonic rodents, so mainly rats and mouse. And basically, the point was when you want to develop neuron in vitro, to order pregnant mice or rats and to collect the embryo at a particular stage, depending on the type of neurons you want to obtain, and then to carefully dissect the brain of the fetuses or the embryo to collect the area that will give rise to the type of neurons you want to study. So, for example, if you see my mouse, we dissect carefully the forebrain when we wanted to model cortical neurons, or the midbrains here when we want to model dopaminergic neuron, and so on. The main problem that we get is that first you need to use a lot of embryo to have a lot of neurons if you have a cell-consuming experiment, meaning that there is a problem of consistency because you will have different pregnant mice or rats and different embryos with slightly different developmental stage that you collect. The other problem, of course, is that the brain of a rodent is quite different from the brain of a human. And more particularly, there is some specific genes in the human that came with evolution, and also different developmental processes and different subtype of progenitors that you only find in the developing human brains. And on top of that, since we wanted to do high throughput screening to find molecules, we need a lot of cells. For those who are not familiar with the process of high throughput screening, it consists in picking some hit compound in collection of molecules that goes from 300 to 100,000 of molecules. Really, the point here is not to select molecule depending on the mode of action or their target, but really to take all the collection and to run an assay, a screening to pick up the good one. And to do so, you need to miniaturize the culture into this plate that contains 384 tiny wells. In each well, you will assess a compound. And of course, it's not humans that are doing the job. It's this kind of automaton that manipulate the plate and will seed the cells and treat the cell with the compound. So of course, when you have to assess 300 or 100,000 molecules, you need a large amount of cells. So it's mainly impossible to dissect enough embryos to have enough cells. And also you see that there is a need for standardization of the culture because you sometimes want to share the collection between subset of compound that you will screen on different days because otherwise there's too much molecules. Or you also want sometimes to repeat your screening with the same compound but on different readout or biomarkers. So standardization is the key. And of course, it's not possible to do so when you're dissecting embryos coming from different females and that maybe have slightly different developmental stage. So that's why I moved first to the UK for my postdoc, and then I joined in 2006 STEM in Avery, because the idea was to use human pluripotent stem cells to produce in vitro human neurons. Of course, in the beginning of the 2000, the pluripotent stem cells that we get were the human embryonic stem cell, first isolated by James A. Thompson. The embryonic stem cell, they are basically dissected from the inner cell mass of IVF blastocyst, the human blastocyst, and the inner cell mass is collected and put in culture. And we were interested in the cells because there are three main properties that were very interesting for us. The first one, of course, they have a human genetic background, so it's important to model the human development. As any kind of stem cell, they can self-renew, meaning that they divide exactly as the same, keeping the same potency. So it's nice in the lab because we thought at that time that we maybe would be able to grow the cells to reach the mass of cells we need for the screening and for the disease modeling. And when we wanted to have neurons, we can use their pluripotency potential, meaning that they are virtually able to give rise to any kind of cell of the body, including virtually all the type of neurons of the brain. And in the meantime, thanks to Professor Shinya Yamanaka in 2007, we also had a new source of human pluripotent stem cell that really changed the game, because he obtained the Nobel Prize for his discovery, because instead of having to collect IVF embryo, which is also a rare resource, now we are able to collect the blood or the fibroblast of any type of patient that we want, or healthy individuals. And by adding 4 transcription factors on the cells, we can induce the reprogramming of the cells, so a drawback in their developmental program, so that they go back to the stage of embryonic stem cell-like, and they are now able to self-renew and to differentiate as neurons. So it was very important for us, because it was like having a patient in a dish. So of course, the main problem that we get is to obtain the good type of neurons, starting from the cell that can virtually do anything. And we spent some years just to find the good cocktail of molecules that we need to provide to the cell to carefully instruct them to first do the gastrulation and neurulation to obtain neural tube-like structure, and then to acquire the regional identity of the brain area that will give rise to the type of neuron that we want to study. So we first obtain the cells to convert into a neural tube-like structure that we call rosette by just exposing them to 2 SMAD inhibitors. And then we pick up the little structure and we expose them to different combination of protein and morphogene, exactly like in the normal development. So for example, FGF8 or Wnt or BMP or sonic hedgehog, to really instruct the cells to have a particular regional identity, so they will produce the type of progenitors that are specific to a neuronal population. And here you have 2 examples of protocols that we develop. And for example, to obtain dopaminergic neurons that come from the ventral midbrain, we expose them to sonic, which is a ventralizing factor, and to Wnt to caudalize the cells. And for the cortex, for example, we do the same protocol, but with different combination of molecules to obtain cortical progenitors and neurons. And each time, since we want standardization and relevancy, we develop protocol to freeze and bank large amount of progenitors that are already committed, so that each time we want to start an experiment, we just have to seed the cell into 384 well plate and to start neuronal differentiation. And basically, this is how they look like. So we also work to miniaturize the seeding of the cells and the process of differentiation as neurons, and to automate it on our big machines. And we finally obtain the standardization of the neuronal differentiation. So here you have some photos of what you can see in the 384 way plates. So at the beginning of the seeding, they look like a progenitor. They form a neural tube-like structure with the good regional identity. After 2 weeks, you start to have the baby neurons forming. So you can see, thanks to the TUJ1 antibodies, the neurites that start to form and to connect to each other. And then after 4 weeks, you can start to have physical synapses that are here labeled with synapsin and PSD95 markers. So all the little orange dots that you can see on the neurite are synapses. And after 6 to 8 weeks in culture, you start to have neuronal activity that you can measure using a microelectrode array or calcium oscillation. And here you have a video of the fluoro4 calcium probe that show you that the calcium is moving, so from cells to cells, and also that the network is beating. So all the neurons are activating in this area at the same time, indicating that there is actually a functional network that's synchronized. And all of this can be detected in 384 well plates by a special microscope or machines. So it's good for the screening. But of course, it was still to make a proof of concept that it actually worked for disease. And at that time, I met Professor Thomas Bourgeron, which is a genetician in the Pasteur Institute in Paris. And he was interested in a particular condition called Phelan-McDermid syndrome, or PMDS. And it's a genetic form of autism spectrum disorder that is caused by the loss of function of one allele of the gene coding the protein CHENG3. CHENG3 is a scaffolding protein that play an important role in stabilizing the glutamatergic receptor at the glutamatergic synapses. And in PMDS, you have 50% less CHENG3 at the synapse, which is the cause of the autism spectrum disorders. And together with Thomas, we go and find Professor Richard Delorme, which is a psychiatrist at Robert Debré Hospital in Paris that have a cohort of PMDS children. And we decide to collect the fibroblasts, the skin fibroblasts from 4 children, convert the fibroblasts into induced pluripotent stem cell, then convert the pluripotent stem cell into cortical progenitor and neurons with a protocol that we designed. And we were very happy when we compare the cortical neurons of PMDS children to healthy individuals. First, to find that actually in our neurons, there were some glutamatergic synapses that incorporate CHENG3, which are the red dot that you can see on the photos. And that in neurons of PMDS children, there is actually half less CHENG3 at the synapse, meaning that really our model is phenocopying what we think happened in the children. And then we use the cells to screen for compound able to restore CHENG3 level and neuronal activity. And we choose as a proof of concept to only screen 300 compounds, but in different conditions, in different concentration and with repetition and replicates, which means that we actually end up with 1,600 experimental conditions. So you can see the necessity to work with 384 well plates and with automaton in this case. And screening the 300 compound, we actually end up with one interesting compound, which was lithium carbonate, which is also known as terlite for the therapeutic name, that was able to restore CHENG3 at the synapse and to improve the activity of the neurons in vitro. And since it's a marketed drug, we feel to give it back to the children that actually provide the fibroblasts for the study. And here, one example of what we obtain with the ASD1, so the first children that we treat. Of course, it was characterized with autism spectrum disorder and intellectual deficiency and due to stop codon on the CHENG3 gene. And when we introduce lithium, we evaluate the social abilities of this child at 6 months and 12 months after treatment. And you can see here on the 2 scale that the result was an improvement into the social abilities of this girl because it was a girl. So, of course, all this data prompt us to perform a real clinical trial that is actually ongoing, and the objective was to recruit 22 patients at the Robert Debré Hospital. 11 were treated with lithium and 11 with placebo, and the objective of this phase 2A clinical trial is first to establish the safety of lithium in this very particular population of children, because first, it's children that they are fragile, and the PMDS children are particularly fragile, and to see if we can also start to see efficiency. And if this trial is positive, of course, the point will be to do a proper phase 2 clinical trial in which we will increase the doses of lithium and really test the efficacy of lithium versus placebo. So how to say that all this story was the proof of concept that the neuron derived from human pluripotent stem cell can be very good tool to model neurodevelopmental disorder in vitro and to perform drug discovery, and also that they will help to reach our goal to go for psychiatric and neurological disorder to a medicine that is more personalized. This is what we call the precision medicine, and that will take into account the diversity of the patient that we have in our cohort. The diversity can be either at the genetic level, the epigenetic level, or the clinical symptoms. So we really think that human pluripotent stem cell can help addressing this diversity. So I hope I'll convince you that with this model we can help improving the 3E in animal research. Regarding marketed drugs, or drugs that are already on the market and that we want to reposition to other applications, we can replace the use of animals for all the steps going from the target identification and the disease modeling to the compound screening and to the selection of good candidates to go to clinical trial. And in this particular case of marketed drug for which the toxicity is known, we can even replace the animal model in the phase of evaluating the toxicity of the compound. Of course, for new chemical entities, it's a bit different. We can still replace the animal in the first step of candidate selection, but of course, we still need animal models to evaluate the toxicity of a new chemical entities on the whole, an entire body. But still, we can maybe try also to reduce the use of animal at this stage by using more complex model. And I know that you will have wonderful speakers talking about the organoids, assembloid, and on-chip technology. So just a quick example of what we are currently doing in the lab. We try to go from two-dimensional neuroglial culture to a 3D structure called organoids. So there are little balls, so 3D structure that recapitulates the development in 3 dimensions of the different cell type of brain area. And for example, here you have what you can obtain by doing organoids of the midbrains. In this case, you can see the development of neurons in 3 dimensions. Here are TH neurons, dopaminergic neurons, but they develop with their companion cell, meaning the radial glial cell, and then the astrocyte and oligodendrocyte. So it's very also nice to have all the cell type together. And we also develop assembloids. So we connect organoids of different regions together. And here you have a video of what you can obtain by connecting midbrain organoids to cortical organoids. In this case, you can clearly see the projection of dopaminergic neurons to the target area. And it will be also very helpful to try to see what's happened to long-range projection and to the activity of the targeted area. And that's it for me. And of course, this work is not done on my own. And I'm really grateful to the people that I have the chance to work with in my different teams, and also all the great collaborators that help us to really convert this proof of concept of using pluripotent stem cell to model neurodevelopmental disorder to an actual treatment, and really to help us to really do a translational work. And that's it for me. I will unshare my screen.
Thank you very much, Alexandra.
And I see you.
Great. That was really insightful, and I think it was setting nicely the scene for this afternoon. So if participants have questions about this talk to Alexandra, feel free to ask them in the chat. I think we have time for a couple of questions.
Yes, there was a question asking if work with iPSC you can only use for genetic disorders or also for diseases which have an early onset and a slow progression.
So of course it's easier with genetic disease because you know exactly what is the trigger. But actually in our hand, we are also using IPS to evaluate risk factor of NDD. It's mainly environmental risk factor. So you can evaluate the effect of, for example, alcohol or for drugs that you expect to or are at risk of producing NDD, for example, the VPA. For multigenic disease or really idiopathic disease, it can be more complicated if the trigger is not how to say, it's not cell autonomous or at least intrinsic to the cell, or if you don't know what to add on top of the cell to induce the problem.
Okay, thank you. And maybe one other question was a question, you used the lithium compound in your first proof of concept study. Why did you choose this one?
Okay, so actually we screened 300 molecules, and on the first criteria that was promoting the transcription and translation of CHANK3 and promotion of the location of CHANK3 at the synapse. And when we screen on this parameter, we only find 6 molecules that were active. When we test the 6 molecules on the neuronal activity, we only find 2 molecules that are actually improving the activity of the PMDS neurons. And we finally choose lithium because it was easier to provide to children to the other molecules. So then the clinician finally chose the lithium among the 2 molecules that came out of the high throughput screening cascade.
Okay, thank you. Do we have time for more questions, or Markus?
I think it would be good to continue now. Thank you very much, Alexandra, again for this
Thank you
insight.
for the invitation.
Thank you very much.